common in fungal called Basidiomycetes, white rot fungi, because these fungi are able to degrade lignin and just release cellulose, which is called white rot. Okay? 20 years ago, when I started working with these enzymes, well, there were no bacterial enzymes at the time described and characterized. Okay, but there was a paper at the time from these Alexström and Zulin where they have applied bioinformatic tools to search for bacterial enzymes based on the sequence of fungal. Okay, so they claim that there are many lactases in the bacterial world, but they were not identified at the time. And I was in 2000, in fact, in the lab of a colleague of mine, Adriano, in my second postdoc. And Adriano was mainly with Bacillus subtilis, which is a bacteria, that gram-positive bacteria, very well known, that produces spores. Okay, so it's a way of surviving nutrient starvation. They produce the spores and they remain dormant and they survive these bad conditions. These spores are extremely resistant forms of cells. Okay? They are surrounded, in the core there is the DNA, and then they are surrounded by a modified peptidoglycan and by a cross-linked protein matrix. And in this matrix, in this protein matrix, there are many enzymes identified, and there is one which is the A, which is COT-A. And at the time I started working with COT-A, that we knew it is a multi-copper oxidase, okay? And we have cloned the gene, and we have expressed the gene in E. coli, and produced the protein, purified and characterized for the first time a lactase from the bacterial world. And these two papers were a kind of seminal in the area, because we were able to characterize, make the biochemical characterization of bacterial lactase. And one interesting property of this enzyme from the spore coat is it's extremely thermoactivity and thermostability. So it's an enzyme that has an optimal temperature of around 75 degrees, and it spends more than 2 hours at 80 degrees. So it's extremely resistant. And short after, we were able to get the structure of this enzyme, which was also the first structure of endospore protein and of a bacterial lactase. And in this structure, we can see the 3 copper-oxygen domains and the copper ions. But as I said, lactases are multi-copper oxidases, okay? So they have copper ions, and they have one mononuclear copper, which is called the T1, based in the ligands, okay, and the spectroscopic features that these copper have, okay? It absorbs at 600 nanometers, and also it has a signal in DPR. And then this is the T1, which is the copper center that oxidizes, take out electrons from the substrate. Okay, so it's the entry of electrons in the enzyme. And then we have the trinuclear center, okay, which is made of 2 T3 type coppers and 1 T2 copper, where oxygen binds and is reduced to water. Okay, so this is the 2 catalytic centers. And of course, for the enzyme to have activity, full activity, it needs to have 4 copper ions. Okay, and this was our first problem when we were producing a protein from bacteria, because we have to produce this, because in fungal most of the lactases are extracellular. Okay, so they have the full complement of copper. But when we want to produce these enzymes in the cytoplasm of E. coli, there are not many copper ions or iron ions inside of, in the cytoplasm of a cell, because they have this activity, metal activity. And our first problem was really to have an enzyme that was fully copper loaded. When we produce the enzyme has 0.5 moles of copper per mole of protein. And when we incubate with copper, we can reach 2.5 moles of copper per mole of protein. So this is really bad because we have a preparation that was copper depleted. And if you want to do, you know, structure function relationship studies, mutations, and all this, it's quite a problem. At the time, I have in my lab this postdoc, Chen, I don't know if you remember him, no, the Chinese, and he was producing the bacteria and in the shaken, in Erlenmeyer shaken, okay? But then he did not book the incubator, okay? And someone came and said, I'm sorry, we have to take out your Erlenmeyer from the incubator because I have to put my things, you know, Chinese, sometimes it's not very by the book. So we take out the Erlenmeyer and put the Erlenmeyer in the bench, okay? And it stays in the bench during the night. In the day after, when he arrived, he looked to the Erlenmeyer and it was bluish. It was bluish. And he said, oh. So he went, I would never do that eventually, but he went on and he purified the enzyme. After the Erlenmeyer being static, without oxygen, okay? Because when we are not mixing, it's without any oxygen. And when he purified the enzyme has the full form of the copper copper ions, okay? So it was really good. So it was, and then, so this is the UV visible spectrum of the enzyme depleted in normal conditions, and after this anaerobic step, after leaving at the bench, it has 4 moles of copper protein. It's totally blue, the protein, and we start doing this procedure always. And then, of course, we read about these homeostasis metal systems in E. coli, and there are literature that support this because in anaerobic conditions, the homeostasis systems that extrude copper outside of the cell are not so effective because there are no oxygen, so there's no Fenton reactions, and cell is more okay with copper ions inside of the cytoplasm. And so it accumulates copper, and if you are overproducing the protein in the cytoplasm, it has the 4. This was really important, and it was a kind of hallmark for the synthesis of these enzymes because it allows us to do good studies based on homogeneous enzyme preparations. We have performed some more studies. For example, the apoform has a K-cut of 1.5, the holoform with 4 complements of copper has, you see, much higher activity, 322. Also, it affected the redox potential, is higher when you have the full copper complement. And also another thing that we have observed is that the haploenzyme is less stable than the holoenzyme. So really, a correct copper incorporation is important for function, for stability. And when we reconstituted this apoform with copper 2 or copper 1, we understood that copper 1 is a better form of copper to restore the activity of the enzyme. And also with the apoform, we could never reach the activity levels of the holoform. Okay, so it's really important that the enzyme is formed inside of the cell. Okay, with copper 1 to have the maximal activity, maximal stability, and we have assessed that by making by following the copper incorporation by UV visible, EPR and Raman, and Daniel was part of this group. I did not put here Raman because it's far from my own knowledge, but we observed that incorporation is T1, T2, and that there are some differences in the T2 when we reconstitute APO and holo depleted variant. So this is just to say that it's important that the enzyme is really produced inside of the cell with the copper ions in order to be everything correctly folded and coordinated and so this is one important thing. And then we went on studying distinct features of this enzyme. For example, one important distinction of bacterial and fungal enzymes is the redox potential. As you know, it's really an important feature of the enzymes, and the bacterial enzymes in general have a lower redox potential, 400, 500 millivolts, whereas fungal have 800 millivolts. And when we look to the T1 center, copper is coordinated by two histidines and by a cysteine, and a difference in the bacterial enzymes is that we have a methionine at slightly coordinating the distance to the copper one, whereas in fungal strains, this residue is a non-coordinating residue, it's a phenylalanine or a leucine. So what we have done, what we used to do in this type of studies is, okay, let's replace this methionine by a phenylalanine and by a leucine and see if we can improve the redox potential. And in fact, we have replaced it by the phenylalanine and by a leucine. We got the structures, everything is well coordinated, so no problem in terms of the structure of the enzyme. It's important. And we have an increase in the redox potential, okay? Not very big, but another millivolt increase in the redox potential. But the problem was that the activity of the enzyme was substantially reduced. And also the stability of the enzyme was, whereas in the wild type we have a clear two-state transition from the native to the naturated to the unfolded state. In the variants, we have an accumulation of an intermediate. So we can replace this residue. We have a higher redox potential, but then we have other properties that do not follow this increase in the redox potential. So we have looked to these type of aspects of the enzyme, and we also look to the mechanism of the oxygen reduction. What we have first performed was soaking of the crystals, of copper crystals, with, for example, peroxide enzyme, which is an inhibitor. We have also have reducing agents. I can't remember if it was the or something like that. But we got the structures of the enzyme fully oxidized, reduced with the peroxide, which is an intermediate in terms of oxygen reduction to water as these intermediates. So we have these structures, and we thought at the time, of course, this was with Peter Lindley, which is a very well known name in the multi-copper oxidase from human origins, from the plasmid, and we have proposed a mechanism for oxygen reduction, and this is based on crystal structures. Okay, so this was a crystal structure of one enzyme, of our enzyme, in fact, because it was the first enzyme where we saw the dioxygen between the T3 copper ions. Then, upon reduction with two electrons, we have the peroxide form, and then upon two more electrons and the two protons, there is the split of the peroxide, the hydroxide between these copper ions and in the copper 2, and then with the incorporation of an additional proton, we have the first water molecule released, okay? And then, okay, a possibility is that there is the migration of this OH to the C4 and a second water molecule that is released, and then with oxygen we start again, or without oxygen we have the reducing form. So, I mean, this is the mechanism for how oxygen interacts interacts with this center without releasing the active oxygen species, which is the important aspect of reducing oxygen in enzymes. Also, in this process, we look to which residues could be involved as proton donors. Of course, there are the protons from the media, but there's a possibility of certain carboxylated residues that are around and part of the hydrogen bond network could be involved in this. And then these were the two that were more close, and we have replaced these residues by alanine, threonine, leucine, by other residues, and they've observed this effect on the activity, and there's a strong impairment in the activity, also in the structure of the enzyme when we replace these carboxylate residues. So, based on these results of cycle-directed mutagenesis, we end up saying that these carboxylate residues are responsible or are important for donation of the protons during the oxygen reduction, and this is also important in controlling the protonation of this residue. So we have been assessing the role of these residues in the mechanism of oxygen reduction. After this work, more structure function relationship work in Cotella case that start to be a bit far away from my own knowledge. Okay? And because I have worked in the past with hyperthermophilic microorganisms, I am a microbiologist as my start. We decided to study other multi-copper oxidases, but now from hyperthermophilic microorganisms. And we went and cloned enzymes from these microorganisms that live at very high temperatures, and we were able to isolate a bacteria from Aquifex aeolix and the Spirobacter aerophil. But these enzymes, although they are very similar to Cotella case, they do not have activity for aromatic compounds. They have very low activity for aromatics. Instead, they are metallo-oxidases. Okay, so they oxidize copper and iron. In theory, it is an example of the study that we have performed with the MCOA from Aquifex aeolix, where you can see that the KCAT-KM for copper or iron is 10 to the 5, 10 to the 6, and for ABTS is 10 to the 4. So it's really much as a higher specificity for oxidizing copper and iron. They are incredibly thermostable with melting temperatures around 100 degrees. And the gene of MCOA is inserted in an operon that is involved in, or as similar, it is with an operon in E. coli involved in copper resistance. So eventually, this enzyme in this organism is involved in copper resistance, in oxidizing copper 1, which is quite dangerous for cells, to copper 2. So should be sure. This enzyme has, not in contrast to lacases, in front of the T1 copper center. T1 copper center is not exposed to the solvent, but instead it has a segment of, in this case, 29 residues rich in methionines and lysine, so it should be highly flexible in front of this enzyme. And in fact, what we have, after all these years, we now know that the vast majority of bacterial lacases are in fact metallo-oxidases. Okay? So the bacillus lacases are lacases in the sense that they have either specificity for aromatics, but the rest, E. coli, Staphylococcus, Staphylococcus, other enzymes, they have either specificity for ions. Okay? So this is what we end up understanding on these for these enzymes. In the case of Cote, we go further, and 2 years ago we had the crystal structure of the enzyme. And when we got the crystal structure, the loop was absent from the crystal structure because it was very flexible. So we need Sachs, Rosetta, and molecular dynamics integration, of course, to define this loop. Okay? And we found out that this loop follows open to close transition and eventually regulating the access to the T1 copper center. Okay? So it can be open or closed because we have analyzed loop truncated variants in relation to the activity with substrates, and we end up, when the loop is truncated, we have increased activity for aromatics, decreased activity for copper ion. So this loop is important in regulating the excess of substrates. And then our idea was, okay, we have an extremely thermostable enzyme, okay, it's like 100 degrees, but the activity is not really good for applications because it is mainly active for time. So let's engineer these enzymes for aromatics. And we have used one technique that we have been developing in these last years, which is directed evolution, okay? So instead of there are two main areas, two main approaches to engineer an enzyme. One is rational, as this to this, and the other is what we call directed evolution, that is based in random mutagenesis followed by screening, like the evolution, the natural evolution in life. So there is mutagenesis followed by screening. Then we select one that has either activity for the substrate of interest, and then we go several rounds until we could improve the activity. So basically, we do error from PCR of the gene, then we transform these plasmids in E. coli. We have a library of variants, could be like 9,000 or something like that. We grow, we break the cells, and we analyze the activity, include extracts, and after analyzing, we select those that have either activity, send to sequence, or we start again. So it is basically cycles of mutagenesis and screening for what we want. Okay, so that's what we have done, and we end up. So here is the wild type. In the first round of mutagenesis, we introduced 3 mutations. Then we mutagenize this variant and screen, and we end up with this, with another mutation here. Mutagenize. Okay, so this is what we have done, and we end up with a variant that shows 100-fold improvement for the aromatic ADPS. What is really good in this engineering approach is that sometimes we really get really high improvements in the activity, not like 2-fold or 5-fold, but sometimes not so reliable. What else we have done indeed? This was another work where our aim was trying to understand. Okay, we have introduced these mutations, and we have done another technique to reduce the number of mutations, maintaining the same activity. But we would like to know what is the role of these mutations in improving the activity. Okay, so and for that we have combined different techniques, X-ray crystallography, from Newton, molecular dynamics, mutagens, and to understand how the enzyme evolved to allocates. And at the end, what we understood is that these mutations, the effect of these mutations were at displacement of residues in short loops close to the T1, meaning it opened up the active site, the T1. Also, the net loop was found more close over the T1, implying that the loop is important for interaction with aromatics. Also, we observed a new substrate binding site, and one important thing, interesting thing, was we by analyzing the network analysis, we have shown that there was a fine-tuning pathway between the mutations that are more distal to the active site. Like if having a mutation far away from the active site has an allosteric effect in the binding site. And this is very interesting because of this. Sometimes we think, okay, I want to improve an enzyme, when we think, okay, we are going to change residues that are close to the active site. It's usually what we do. But but in guiding evolution, some mutations were very far away, and we proved that these mutations, not only as have proved that these distal mutations can affect dynamics of the protein and affect regions that are far away. Okay, this was very interesting in this sense. Okay, at this point of my work, I already have a mission to my lab. Okay, so and I have started working with other enzymes, but our mission was identify and characterize new lignolytic enzymes and auxiliary enzymes that help lignocellulose valorization, in particular, because most of the people are targeted in cellulose, as you know, because cellulose is really a good substrate for everything, or any cellulose, because they are sugars. But our target was more the lignin. Okay, so we want to identify enzymes that somehow modify lignin, degrade lignin, valorize lignin, and we started analyzing the characteristics of our bacterial enzymes in comparison with fungal enzymes. Again, and one thing that we observed, it is the optimal pH. The optimal pH this is a fungal enzyme, and this is our enzyme, and what we have observed is that our enzyme and all bacterial enzymes, all bacterial lactases, have optimal pH for phenolics in the neutral to alkaline range, whereas the fungal have optimal pH at around 3, pH 3. And the reason for that, and the optimal pH is at it's a pH close or behind the pKa of the substance, meaning that the activity of the enzyme is higher when the phenolic is deprotonated. Okay? So as the pH increases, there are either population of deprotonated compounds is higher, the redox potential is lower, and the enzyme is higher activity. Okay? So higher activity is associated with the protonation of the phenolic. For the fungal, the reason behind optimal pH in the mechanism is alkaline and acid in the acidic range is because all fungal enzymes have an aspartate close to the T1. T1 is here, the aspartate is here. That helps, this is the phenol, that helps stabilizing the phenolics and the phenolic radicals that are formed. Okay? And these bacterial enzymes do not have this aspartate, so they only rely on the equilibrium of the substrate in order to optimally take out electrons from the substrate. I don't know if this is okay. So this was one thing. Other thing is, if you want to address lignin, only 20% of lignin are phenolics. The other 80% are non-phenolics. And usually to oxidize or to modify the non-phenolic part of lignin, the non-phenolics are not substrates of lactases, okay? Not non-substrates. We use mediated reactions. Okay, we have mediators in order to get the enzyme mediators and then oxidize the non-phenolics. And one possibility is to use these phenols as mediators, and we have tested that. So we have the lactase. We want to oxidize vegetryl alcohol that we yesterday was talking. The vegetryl alcohol is not a substrate of lactase, but if we have a phenol, this phenol is going to be oxidized, and it's going to reproduce a radical that then interacts with these compounds, that could be many, and then you have oxidation of the. One thing that we learned from this is that if we want to choose a good mediator, it's not the higher activity. Okay, so we tested 3 different phenols. We have single aldehyde was the best substrate, but the best substrate is not the best mediator. Okay? Why? Because we need reactivity, but we also need stability of the radicals. Okay? And in this case, what we saw was that methylseringate, which was the worst substrate, is the best mediator. So maybe this makes all sense to you, but that's what I remember. So another thing that we saw on this work was that we need to add high amounts of mediator in order to have higher conversions of this to this. And for example here, the ratio substrate mediator, no, mediator substrate, okay? If we increase the ratio mediator substrate, we have higher conversions of the non-phenolic, okay? And this is related because I don't know, there are thousands of papers and you read that and there are people that say, oh, these are not mediators, these are enhancers. But at the end, what the thing is compounds that we are using as mediators, these phenolic compounds, they are involved in this reaction, but they are also involved in many other reactions. And we were able to identify coupling compounds that were produced. So the thing is, when we are using these substrates as mediators, okay, as there are many competing reactions, we need to have higher amounts of mediators in order to have higher conversion rates. Okay, this is more on the applied side, on the how to understand what's going on this reaction. Another topic, and I'm almost finishing, okay, another applied topic that we have focused was on decolorization of synthetic dyes. Okay, so you all know that there are thousands of dyes in the world, and that the dyes are synthesized to be robust, to be non-degradable, and then we want to degrade them, and there's no system to degrade these dyes, and they are released into the wastewaters, and there are certain pressure to set up the biological system and the enzymatic system. So we have tested our Cotella case at the time for the decolorization of a range of dyes. And as you can see, there are dyes that are less, dyes that are more decolorized. And we have tested, we have studied the decolorization of two dyes, one azodite and one anthraquinonidine. So these were two papers that we had. With the azodite, we have done many things, but we followed the decolorization. As you can see, the dye, which is orange, turns brown. And also, after centrifugation, you can separate solid. Okay? So this was an indication that polymers, oligomers and polymers were produced. And in fact, based on this was in collaboration with a colleague of mine, this Aula, which is a chemistry. She has, we have proposed a degradation pathway that end up in the production of radicals, and these radicals that are produced are able to, to interact, react with the dye itself, with the intermediates, and we were able to, by Maldiv and mass spectrometry methodologies, we are able to identify several oligomers that were produced. So in this case, the decolorization was this reaction, okay, this coupling reaction, this production of radicals in this oligomer production. At the end, it's really nice because we were able to see that there is a detoxification, okay, because it was out of solution. In the case of the other anthraquinone, what we saw was a conversion of the blue into a red. So it was not a decolorization, but it was the production of another type. But there are people that claim this is a biologically synthesizing dye. So again, sorry. this is the dye that accumulates in higher amounts, in really high amounts. So this was the purification. And in this slide, I'm just presenting. This was a PhD student that I have with my colleague, the chemistry colleague Paula Cabal, where what they have explored was the oxidation of aromatic amines, okay, for the production of many different compounds. We have many papers, but in chemistry, there are no papers. In this case, for each compound, we have one. So we produce several interesting products, biological active products like phenoxazinones, phenazines that are at the basis of many antibiotics or other molecules, interesting molecules, also azo compounds and the dyes to color the hair. So it was very interesting, this collaboration, because it allows me to make sense of my work, to understand if we can apply our enzymes in something that is as valid as interesting. So I tried so at the end, in these 20 years, I was able to define with this work and others, I was able to define what we do in my lab, which is, as Daniel mentioned, select new enzymes, and we are mainly focused on oxidoreductases. We aim at advanced understanding of these enzymes at molecular level. We want and we are able to improve the performance and the robustness relying on experimental and lately also in computational tools, computational engineering tools. Also set up this enzymatic process for valorization of bio-wastes and renewable feedstocks. Okay, and at the end, what we want is new enzymes, new bioprocesses, new synthetic schemes, new knowledge, new topics of research, new funding, new students. Okay. And the enzymes that we started, as I said, with the lactase and tallooxidases, and then we also worked with DIP type peroxidases, pyranose oxidases, galactose oxidases. We still did not publish this work, but we are also these flavoreductases, azoreductases that are important for the polymerization of the dyes, that are really good in reducing the azo bond to aromatic amines. And also this enzyme, isogenol monoxygenase, that converts isogenol into vanillin. So isogenol from lignin to vanillin. So we have been studying this, and this is the funding. These are the unit. At ITTB we have these research units which I'm not sure. And this is Fundação para a Ciência e Tecnologia, and we have also some funding from Europe, already 4 different projects, and also a project with Brazil, Ciências Sem Fronteiras, and these are the acknowledgements that because so in ITKB and outside of ITKB that have people that have contributed to the work at ITKB. We have there are many groups that are devoted to the study of enzymes, proteins, like spectroscopic techniques, X-ray, structural, biophysical approaches. It's really good to collaborate with them. Also, we have research facilities that help us in producing proteins or refined proteins, this type of things. And these are my students, many students over all these years, and this Magali there. And that's all. I hope it was not too, it was okay, not too long, but did you know any questions? Please.
Thank you very much.
It's there.
Nice body of work, collaboration. Regarding the common method where we made the synthesis, so lactase and all these dyes
here.
That's one. One, two.
Last one. The last
No,
one.
it doesn't have a huge number, not this low.
Sorry. Okay, I thought it was a I'm sorry, yes. In this case of the green
I can see only
I'm
the
sorry.
green.
Okay, yes.
Okay, you have all these components. How do you compare? I know that is, I understand that this is a cleaner way to see disaster. But what about the cost and the easy to make of this stuff?
And this is not it.
But about
But I can
the
tell
workout of the reaction.
What I can tell you is that the yields are really high in majority of cases. But I remember that Paula used to write very good to excellent yields. So it was above 80%. So, but of course, I don't know how this compare with other. I'm sure it doesn't compare very well. Because the question of using enzymes is not the cost, it's more the friendliness of the process. It's more on that. Eco-friendliness. That's what people think. Because on the cost, it would be difficult to beat chemical. Yeah. So we need like policies that, this is not possible. So even if it is very expensive, you need to do other way. Exactly. It's green.
Before discussing the selectivity, the sense of different applications of substances, metals, or different types of substrates, if I understood correctly, you are putting all the weight of the selectivity in the binding sites, either the structure, or the flexibility, for instance, if this aspect of the series there or not, stabilize the radicals, or the this cover, kind of cover, opens or explodes. But what about the metal centers? Are there differences?
Because the thing is that this is something very interesting among the cases is that they are almost indistinguishable in the metal centers. When you compare the structures, they are equal. So we cannot understand. This is a question. This is the question. How glycases that are indistinguishable have so different specificities and rates of reaction? That's what we want to understand. It's one question on these enzymes because the copper centers, for example, the T1 with the distances, the coordination, everything is equal. So the only thing that is different is exactly these second shell residues, this surface close to the T1, but it's even this is one thing because when we, for example, when we have this values at 100 times higher activity, I was expecting, well, I'm going to look to the crystal structure, it will be clear why. No, it's not clear at all. They are the same. You have to do this molecular dynamics and see the dynamics of the protein. It's really interesting that aspect. So it's dynamics of the protein. It's not the structure, it's not the crystals, it's not where they are, it's how they behave.
They have Solomon and Friends call the static effect. It's a fixed structure, no matter if the metal is there or is not. It's like this, because this is a common, something relatively common in mononuclear nuclear sensors. I don't know in the case of this new sensors. We can also talk about the static effects. So it is different the structure of the protein with or without metal.
Well, from my, in my view, there's no difference. Because we already analyzed without and with, and the in that report that we have performed with you, what we saw, the differences we saw are very, very small differences. We could see differences in, because we can follow proper interpolation, doing the transient state kinetics, and we see that it's very, it's not, for me, it's not, I mean, because I'm not a biophysical person like Solomon that knows about the electrons and this kind of things. At this level, you can have some differences, but
this is a single breakup of that thing. Structures are the same. Willow without metal, then Solomon is right.
yeah. Well, yes. No, but then the thing is, for example, if you produce the apoprotein, or if you start with the Aula, deplete the enzyme, and then start, there are differences, okay? So it seems that there are some, so you can produce the apoprotein with Aula, or you can produce the apoprotein, and then deplete the apoprotein with EBTA, and then start with the, reconstitute the apo, but, because of all, and there is differences. In fact, you see that.
Inactivity, yes. It's like
Yes,
belief.
inactivity,
It's like belief.
but that's what I want. Yes,
Yeah.
that is good.
Congratulations on your talk. It's amazing. Before doing my question, I wanted to acknowledge you because I was working with the bacterial package and I couldn't get the blue glove cover until I read your paper. And so I tried these microaerobic conditions and it worked. I was
Exactly.
so excited. Yes, you said all our life.
But we never wrote this story of Delta Meyer in the bench.
But well, then I wanted, I have a question about the directed mutagenesis. So you mentioned that you obtain mutants with higher activity towards aromatic compounds. But I would assume that these mutations also affect other properties like stability or optimal pH or other things. Did you check anything?
We check everything. And in general, we saw differences only on the activity.
Okay.
Yes, not in the open. General, it's only the activity that, okay, there are sometimes some fluctuations, okay, of the optimal is not 4.5, it's 5.2. It's not nice. It's not really, but you always check it because if you want to assess the role of mutation, you really need to do disease in these kind of things. But of course, there are different things.
And you could do direct mutagenesis to increase product potential, for example.
We never, even now with the MCOA, because we are now with the MCOA, we saw that when we change one residue that is close to the trim, well, in fact, it's adjacent to a ligand of the T1. And when it is glutamate, and we have, no, it's an histidine, and we are changing to this glutamate, and we have an increase of activity to granulate. It's one of that I said to you. And then we observe that it goes to the surface, goes to the tube, and there are several carboxylate vessels, two glutamates and one aspartate. And if we are moving for activity for ABS, which is negatively charged, if we take out the charge out and it increases glutamate, which take 10 seconds. And this we asked Professor Flip Coswell to why again, because we have this improved connectivity and we look to then we have the disclosure and they don't see any difference at our eyes. with our methodologies. And we asked to check the level of expectation because initially the level of expectation is exactly the same in different. So we can never really increase the level of expectation, even when we increase the activity, but brutally, and the level of expectation is the same. So it's other things, but what they do, I don't know. So it's my limit, because then it goes to the biophysical.
Probably because you have this, you have this fact for a time. If you make a non-conservative mutation, it's non-conservative enough, assume the word expectation, you're fully over the structure. Because actually, in T1 plasmids that do not have the center coupled, take this Thailand orders, if we can assume the potential a lot, and this is like one whole. Yes, it's amazing. But of course, you have the freedom to move this without disrupting anything else.
Yes.
It's analytically active. And this gets removed there, which has the gate, the electrons. Here. These two sensors are linked. Via the Thailand.
This is complex because we have this volume and yes. Okay. This loan is only in because, for example, in NCOP from Pirovac, there's also lupus that is 13 resolutions. In Nikolai, the loop is also very long, 40 resolutions, but it also depends on the defined loops. Because, for example, in Nikolai, in the crystal structure, they have the loop. They were able to see the loop. In our case, it is so flexible because it's 29 and you have 12 glycines that it's not in the structure. But all these bacterial enzymes have the loops with some islands, some more or less.
Maybe I misunderstood, but you said that there's no changes the root and both present was equal for the subject aromatic to enter the property.
Decrease the activity for the matrix and decrease for the I cannot explain.
Yeah, that's why I said,
But sometimes if we go too far in explaining, it's really strange.
it's been years,
Yeah.
that's why
There are
I
some,
asked
no, there are many,
you.
yes, I know, but there are some papers that mention the interaction of methionines with aromatics, which is something that is already described, that there is some affinity.
Another question? No?
Thank you.